Welcome. In this session, I'll continue discussing about pest resistant crops. So in the last session, I discussed about BT crops. Now in this session, we will see about how RNA interference is used to develop pest resistant plants. Then thereafter, we'll also start discussing about biotechnological applications in medicine. So let's begin this session with the pest resistant crops. So here under this we will see about RNAI. So by using this RNAI mechanism, we developed pest resistant crops. So here there is a nematode, so whose name is Meloidygyne incognitia. So this is a parasite. So it's a nematode parasite, and this one is going to infect the roots of several different species of plants. We'll see in this case tobacco as an example. So infect roots of tobacco plants, and because of this the plants will not grow well, and there is a huge losses to the farmers. Okay, so this Meloidygyne incognita, right, infects these roots of tobacco plants, and it is a nematode parasite. Okay, so it infects the roots, and it takes away the nutrients so that need to be supplied to the plant, and because of this the plant will not be growing properly, and there is a huge loss to the farmer. Okay, now by using this RNAi mechanism, right, we can stop this Meloidygyne incognitia to grow in the roots of tobacco plants. So now how they have done it, so let's see first this RNAi mechanism. So here this RNAi naturally takes, so it naturally takes place in all eukaryotic cells. So it naturally takes place in all eukaryotic cells, right, as a part of cellular defense, as a part of cellular defense. Okay, like in case of viral infection, so to stop that viral infection, so we know that once the virus enters into the host cell, its genome will get integrated into the host genome. Okay, and then it starts getting expressed and it starts producing the viral proteins, and it will take control of the host cell. Okay, so in controlling such viruses, okay, so these eukaryotic cells have developed something called RNAi mechanism, right, from molecular basis of inheritance. So where we have some basics. Okay, so here we know that the process of transcription takes place inside the nucleus. So this is the cell and this is nucleus, and inside the nucleus, right, from the DNA the process of transcription takes place, and it results in the formation of mRNA. So this is mRNA, and this mRNA will be transported into the cytoplasm. I hope you remember that the transcription in eukaryotic cells will take place inside the nucleus, whereas the translation takes place in the cytoplasm. Right. Whereas in case of bacteria, that is not the situation. Both transcription and translation will take place, okay, at the same place, that is cytoplasm, as there is no nucleus in case of prokaryotic organisms. But in eukaryotic cell, transcription is within the nucleus. So once this mRNA is generated, it will be transported into the cytoplasm. So here this is the mRNA that has been transported into the cytoplasm, where okay, it undergoes translation. So for that purpose, ribosomes need to come and attach and the tRNA molecules, and they are going to read out the information present on the mRNA, and according to that they are going to join the amino acids and make polypeptides. Now if I am introducing okay a complementary sequence to this mRNA, so we'll see that one here. Okay, say suppose this is the mRNA molecule, and on this mRNA molecule the sequences are something like this: A, G, C, U, C, C, G, U, A, some sequence we see on the mRNA molecule. Now if I'm going to introduce a complementary strand to this mRNA, so what we mean by this complementary strand, so which will have complementary sequences. So in this case we know A binds with U, G binds with C, G, A, C, G, G, and C, A, U. So this is complementary RNA molecule. Say it's a complementary RNA molecule. Now when we introduce this complementary RNA molecule into the cytoplasm, what happens? It binds with this mRNA, right? And it becomes a double-stranded RNA molecule. Okay, so this will result in the formation of a double-stranded RNA molecule. If the double-stranded RNA molecule is formed, then this mRNA cannot be translated. Okay, so here the double-stranded RNA cannot be used in translation. So if translation doesn't take place, the polypeptide that need to be formed from this mRNA will not be synthesized. So I can say that we have stopped the expression of that gene. So here we know this mRNA has come from a particular gene, okay, from the DNA, okay, and it codes for a particular polypeptide, and we have stopped that mRNA to undergo translation. As a result, that protein which need to be formed from this mRNA will not be formed. So which I mean that we stopped the expression of that particular gene, right? So in this way, right, we can stop the expression of the genes. Now whenever we find that double-stranded RNA molecules inside, so this is the complementary RNA molecule, and this together it becomes something called a double-stranded RNA. Now the presence of the double-stranded RNA activates some other enzymes, and they are going to break this double-stranded RNA into fragments. Okay, whatever it is. So here this double-stranded RNA will never undergo the process of translation. So that's what the RNAi, so which stands for RNA interference. So here the full form of this RNAi is RNA interference. As we have stopped the expression of the gene, I can also call this as okay the gene silencing. I can also call it gene silencing, right? Okay, so instead of gene silencing, I'll take RNA silencing, because that word gene silencing is a broad term under which this comes RNA silencing or RNA interference. Okay, so this is how the RNA interference or RNA silencing, so we are not allowing the RNA to undergo translation. Now, how this RNA mechanism has been used, okay, in developing the pest resistant plants, let's see. So scientists, what they have done is they introduced the nematode genes into the tobacco plant. So what they have done is, whatever that parasite specific genes, so they introduced into the host plant. So that is, here we have taken the tobacco plant. So here parasite specific genes introduced into the host plant. Okay. Of course, that they have done it by using the vectors. So that is the plasmids from a bacterium called the Agrobacterium tumefaciens. So the vectors that we have obtained from this bacterium, so that is Agrobacterium tumefaciens. So we know that already in earlier sessions we studied, we took that vector, and this vector from this bacterium, Agrobacterium, is very good at delivering the gene of interest into the host organism, especially in case of plants. So the vectors that we have obtained from this Agrobacterium have been used to deliver the nematode specific genes into the host plant, and the introduction was in such a way that the host plant is able to produce, okay, the parasites genes, so which will be having RNA, and also it's a complementary RNA. So both are produced within the host cell. So here host cells, that is the tobacco plant here. So in this case, so they are able to produce mRNA, and also its complementary RNA, So both these are going to actually be produced within the host cell. So that introduction, so how we have made, so the transcription of the normal mRNA molecule and its complementary, okay, RNA will also be generated by the process of transcription. So when there is a mRNA, there will be complementary RNA also that will be produced, and both of them will have complementary sequences, and they are going to bind together and result in the formation of double-stranded RNA. Okay, right. Of course, here there is one point I would like to again mention: the introduction of this complementary RNA. So, the introduction of this complementary RNA into the host organism. So there are different methods. So we use different methods to introduce that complementary RNA into the host organism. So that is, okay, one by infection with viruses. So infection with the viruses that have RNA genome. So that have RNA genome. So that you can do it. Okay. So that complementary RNA. So because we know that in the earlier sessions we studied that even the viruses that can cause diseases, you can make them non-pathogenic or disarmed, and then use them to deliver your gene of interest. So you can make infection with viruses that, okay, that have RNA genome, right? And that can even happen naturally. So not only by us, it can also happen naturally. Then, okay, or by mobile genetic elements. So or by mobile genetic elements, okay, that replicate via RNA intermediate. So that replicate via RNA intermediate. So here these mobile genetic elements are called transposons, So here in our earlier sessions or in previous sessions, you might have understood that there are genes which are located on chromosomes, and you might have thought that the genes have specific locations on the chromosomes. I mean, those genes are fixed on the chromosomes. So whenever you see a chromosome, okay, we already identified the locations of many genes on the chromosomes. Say some gene X is present here, then after that some other gene. So these are different locations of different genes, and we thought that these genes, okay, not all, some of the genes uh, have ability to relocate from that, or within the chromosome. So that's those are called transposons. So transposons are the genes that relocate, so means change their position. Of course the other genes may be fixed, but there are certain kind of genes, right? So these are all the genes. So we cannot take into account that all genes are fixed on the chromosome. So there are exceptions for this. So there are certain kinds of genes that don't have a fixed position. They can change their, okay, locations, and those genes are called the mobile genetic elements, or in simple terms they are called transposons, right? So that's the source of the complementary RNA. So here in this case, what we have done is whatever that host specific, parasite specific genes, these parasitic specific genes are introduced into the host plant, of course, by using that vectors from the bacterium, okay, agrobacterium, and the introduction was in such a way that the host cells are able to produce the mRNA and also its complementary RNA. And when these two are present inside the cell, it results in the formation of double-stranded RNA. And presence of the double-stranded inside the cell, it triggers RNAi. So it triggers RNAi. So what happens is this mRNA will not be allowed to undergo, the polypeptide, whatever that polypeptide need to be formed from that mRNA will not be formed. So usually these parasites, after entering into the roots, they will be converting some of the cells and use them as feeding sites by secreting some kind of substances. Now, if we are able to stop that secretions from that parasites by using this RNAi mechanism, then those host cells cannot be used as a feeding sites by these parasites. As a result, they will not be able to get the nutrients, they starve, and then they die within the roots. So that's what how we have developed the pest resistant crops by using this RNAi mechanism. Right, so that's all about the biotechnological applications in agriculture. So how we are using biotechnology in agriculture. Next, we will see about how we are using biotechnology in medicine. So the second one, so biotechnological applications in medicine. Biotechnological applications in medicine. Okay, so here uh, we have I mean, by this time already a lot of advancement have taken place in medicine by using biotechnology. Okay, and with this we have developed around 30 recombinant therapeutics. Okay, so 30, okay, recombinant therapeutics are already present in world. Okay, but out of these 30 recombinant therapeutics, so only 12 are being used in India. So out of this, only 12 are used in India. Of course, all these 30 recombinant therapeutics are for human use. So for human use, so they have been approved. Okay, out of that, only 12 are used, okay, India, and one of that is this insulin. Okay, so that is called genetically engineered insulin. Genetically engineered insulin. I hope we know that this insulin is a hormone, and this hormone reduces the blood sugar level. So hence we call it the right hypoglycemic hormone. So this insulin, so we consider it as the hypoglycemic hormone. So it reduces the sugar level in the blood. Okay, now there are certain conditions, that is diabetes, the diabetic patients, where they require this insulin so that to reduce the sugar level in the blood. Now, in earlier days, the people who used to suffer with this diabetes, they used to get insulin that have been extracted from animals like cattle, pig. They used to slaughter them and then, okay, take out the pancreas and extract insulin from those animals. And that insulin that has been extracted from those animals is to be injected into the humans. But the point is that those insulins have different structures when compared with the human insulin. So there are slight changes in the structure of those insulins which we obtain from other animals. As a result, whenever we injected these, you know, insulin that is obtained from other animals, some people used to show some reactions towards them. So hence it is necessary, it became necessary to make insulin that exactly is similar to human insulin. So how is that possible? So biotechnology made it possible by isolating the genes that code for this insulin and introducing that genes into a bacterium and make that bacterium to produce insulin. So that's what I'm going to discuss under this genetically engineered insulin, right? So here first we will see that the normal structure of this insulin. So this insulin has around 51 amino acids. So it has, okay, total of 51 amino acids, and this insulin is made up of two polypeptide chains. Is made up of two polypeptide chains. One is a alpha chain. So you will see, so this is an alpha chain, and another one is a, okay, beta chain, right? So this is alpha chain and this is beta chain. You can take alpha chain as A chain and beta chain as B chain. So this alpha chain is made up of 21 amino acids, whereas the beta chain is made up of 30 amino acids. So it's made up of, okay, alpha 21 and beta 30 amino acids, and these two chains, okay, are linked by, right, some disulfide bonds. So they are linked by disulfide bonds. So this is how we see in case of a mature insulin. Okay, of course, when it is produced, it will be not in mature insulin. So the human cells, that is pancreatic cells, the beta cells of islets of Langerhans of the pancreas, so they have mechanisms to make this insulin. Okay, I hope we all remember that this insulin is produced by the beta cells of islets of Langerhans. Islets of Langerhans, and the cells are specialized to construct this insulin. Okay, of course, when it is prepared, it will be not in this form. So this is A chain, B chain. It will also have some extra polypeptide chains that have been removed, okay, means trimmed, and then whatever, and made with only alpha and beta chains linked by these disulfide bonds, and this becomes a, right, mature insulin. So that's what I'll mention here. So a mature insulin that is ready to function. Okay, and one more point, the mature insulin, which means it's a functional insulin. So this mature insulin is a, functional hormone. It's a functional hormone. So this whatever insulin, when it is produced, it is produced in an inactive form, and that is called the proinsulin. It's called proinsulin. So this is a inactive or non-functional hormone. It's a non-functional hormone. Okay, so when we see this proinsulin, it will be having, okay, a structure. So that is saying something like this. Okay, and you might see a color. Okay, I think in the textbook they have mentioned something with a green color. So that one is a beta chain or B chain. So this is green in color. So if you don't think that this is green, okay, here just I'm referring with the textbook and telling you to identify the colors. In the textbook they have given the diagram with the three different colors to tell you that, okay, there are three polypeptide chains, okay, attached one to one. Okay, and this green colored one is the beta chain, and the blue colored one, so this will be the blue colored one, and this is the alpha chain or A chain, alpha chain or A chain, and the remaining, that is brown in color, so this is the brown in color, this one is the C chain. Okay, so A or alpha, okay, you can say it's a gamma chain, and this is B or beta chain. Now this beta chain will be eliminated, so I'm sorry, the C chain will be eliminated and only the beta chain and alpha chain will be present. So C chain is eliminated, so that's been removed, and once this is removed, the alpha or the A chain and beta chain will be linked by these disulfide bonds, and this proinsulin will become insulin. So the proinsulin is going to become insulin, that is the functional hormone. Okay, now how they generated this insulin by using bacteria. So bacteria don't produce insulin, so hence it's a heterologous host, right? So scientists, what they have done is they have isolated the genes that code for the B chain and A chain. So the genes for A and B chain were isolated from that beta cells of islets of Langerhans. And now what they have done, so those genes they have linked to the plasmids of E. coli bacterium. Okay, say this is the gene that has been linked. So say it is coding for A chain, and another one with the B chain. So B chain gene, and this is A chain gene. Clear this one? So once they have linked to this, so this is the plasmid of E. coli. So this is also the plasmid of E. coli. See, they have prepared separately, A chain separately and B chain separately. And now this plasmid that contains the gene for A chain is inserted into the bacterium. Okay, so this is transformed, okay, with the plasmid having the gene for A chain, and again in this case also same. So this is the gene that will be having the beta chain. So that plasmid having the beta chain and beta gene. So this one with the A gene and this one with the B gene, right? And once they are present inside the bacterium, they are going to get expressed, and as a result they are going to produce the A peptides and B peptides, which will be collected later. Of course, you may be asking, so where they will be growing? All this we will grow them in bioreactors in large quantities. Okay, and we're going to collect in large amounts the A peptide and B peptide. So this will give the A peptide, and this will produce the B peptide. So that's going to give the B peptide, and those two will be joined by using a chemicals that can form the disulfide bonds between the A chain and B chain. And this was made successful, okay, by Eli Lilly company. Okay, for the first time they synthesized the insulin that is exactly same as that of human insulin. So there is nothing different, because we have taken the gene that codes for the insulin from the humans only. So hence there will be no differences at all. It's just a clone. So now when this insulin is introduced or injected into the humans with that diabetes problem, it's not going to create any kind of side effects. Okay, so Ali Lilly company in the year 1983. So in the year 1983, so they have successfully developed, synthesized insulin by using bacterium. Okay, so made bacterium to produce human insulin. To produce human insulin. So this Eli Lilly is a American company. It's an American company, okay, and they made it for the first time successful, okay, in the generation of this human insulin. Right, so with this I will end today's session. In the next session we'll see about Gene therapy. Still, I need to discuss many more applications of biotechnology in medicine. So another application is the gene therapy. Then later we'll also see molecular diagnosis.